This is part 4 of the 2.4 notes on macromolecules, and this is our final macromolecule, the nucleic acid. Nucleic acids are long macromolecules made from carbon, hydrogen, and oxygen, like all the other macromolecules, but also nitrogen and phosphorus. Here we're introducing a fifth element, that is phosphorus. The main function of the nucleic acid is as an information storing molecule. So information about living things are stored inside of these nucleic acids. Nucleic acids are the molecule of heredity. We'll talk about genetics and heredity later on in the year, but simply, if you look at a parent, those parents have characteristics that they can pass down onto their offspring. This is done through the action of nucleic acids. Now, you're probably familiar with the main kind of nucleic acid, and that is DNA. DNA has a partner, and we call that RNA. The initials DNA stands for deoxyribonucleic acid. So the fact that it is a nucleic acid is right there in its name, deoxyribonucleic acid. RNA is ribonucleic acid. These are the two main kinds of nucleic acids, and they both serve similar roles, but they are slightly different in structure, and we'll talk about that as we go along. The building blocks of nucleic acids are nucleotides. Nucleotides are the monomers that are used to build long nucleic acid polymers. These nucleotides have 3 subunits as well. So each nucleotide is made out of 3 components. We have a phosphate group, which is a phosphorus atom surrounded by a bunch of oxygens. Then we have a 5-carbon sugar. These are sugar molecules that have 5 carbons as part of their chemical structure. And then we have what is called the nitrogenous base. Each nucleotide has a nitrogenous base, and different nucleotides have different nitrogenous bases. If we look at DNA, DNA has a very familiar shape, and this is called the double helix. We can kind of describe the double helix like a twisted ladder. So if you imagine you have a ladder with the runners on the side, and then you have the rungs, the steps going horizontally, you take that ladder and you twist it, you get this double helix shape. Within the nucleotides of DNA, the 5-carbon sugar is called deoxyribose. This is where the first part of the name deoxyribonucleic acid comes from, is this deoxyribose sugar. There are 4 different nitrogenous bases that are a part of a DNA molecule. This is adenine, thymine, cytosine, and guanine. We're going to talk a lot more about DNA and how it's put together and how it works later on in the year, but a very important principle about these nitrogenous bases is they always pair together in a very particular way. Adenine always matches with thymine, and cytosine always matches with guanine. And these are very commonly abbreviated by the first letters. So A always goes with T, C always goes with G. So the latter shape of the DNA molecule comes from the fact that the phosphate groups and the sugar group, the deoxyribose, creates a backbone. And this is often called the sugar phosphate structure or the sugar phosphate backbone of DNA. Then the nitrogenous bases act as the steps of the ladder. Each nucleotide matches with a complementary nucleotide on the other side of that ladder. The nitrogenous bases connect to each other through hydrogen bonds. A really important structure of the DNA molecule is the nitrogenous base pairing, and this is often called base pairs. So adenine goes with thymine, cytosine goes with guanine. And the reason why these nitrogenous bases always pair together in this way is because of the hydrogen bonds they form between the two. Adenine and thymine are looking to form 2 hydrogen bonds to connect together, whereas cytosine and guanine are trying to make 3 hydrogen bonds. If adenine tries to match up with cytosine, they won't fit together because if you have 2 bonds and 3 bonds, they won't sync up together properly. So we always get the same base pairing, A with T, C with G. There are 3 major differences between a DNA molecule and an RNA molecule. The first is that RNA is a single strand rather than a double strand. So RNA is only half of that latter structure. The next difference between these two nucleic acids is that RNA is made out of a 5-carbon sugar called ribose instead of deoxyribose. So this is the ribo of the ribonucleic acid. So the difference between these two sugars is actually pretty simple. Deoxyribose versus ribose, if you listen carefully to the name, deoxy is in reference to an oxygen atom. So the deoxyribose is actually missing one oxygen atom that the ribose sugar has. So by changing those sugars, we cause those molecules to behave in a slightly different way. The final difference is that RNA does not have thymine. There are no T's in RNA. Instead, we replace that with a nitrogenous base called uracil. So anywhere in RNA where you would have a T, instead you put a U. So again, the three major differences between RNA and DNA. DNA is a double strand, whereas RNA is a single strand. DNA has deoxyribose sugar, and RNA has ribose sugar. And finally, DNA has adenine, cytosine, guanine, and thymine as its nitrogenous bases, and RNA has adenine, cytosine, guanine, and uracil. So the thymine is replaced with uracil. It's important to remember these three differences because a question about that will definitely show up on your next test, and it usually shows up on the EOC as well. When we look at nucleic acids, we are talking about your genetic code. So what do we mean when we say your genetic code? Well, we're talking about the sequence of nucleotides that makes up that long chain of the nucleic acid. Essentially, each letter represents a piece of information, and when you chain those letters together, it gives us a code that tells the body something it needs to know. So the sequence ACGT, ACGT could be the code for a particular piece of information that the cell needs to know. We're going to get into a lot more information about how DNA works later on when we talk about genetics and heredity. But essentially, DNA is the instructions to run the cell. Everything that the cell needs to know about how to build a cell and how to operate the cell is coded into that DNA. A question might be, why do we need both DNA and RNA? It works out that DNA is the permanent copy of your genetic code. In eukaryote cells, that DNA is always found inside the nucleus. But when it's inside the nucleus, it can't send instructions out to the rest of the cell. So RNA is the working copy of your genetic code. So if we imagine the cell as like a factory, the nucleus would be the office of the manager of that factory. So when the manager needs the workers in the office to do a particular task, they're going to send a message to that factory floor. Now, we don't want to send out the permanent copy of our instructions. We want to send a working copy. So kind of like making a photocopy of a page out of a textbook, it doesn't matter what happens to that photocopied page because we have the textbook left behind. So what the nucleus does is it makes a copy of the instructions on the DNA into a form we call RNA, and then that RNA copy is sent out to the rest of the cell to perform a particular task. If that RNA copy gets damaged or broken or breaks apart, it doesn't matter because we still have a permanent copy within the DNA. Now, we've only briefly talked about what nucleic acids are and what nucleic acids do. Again, we'll spend a lot more time talking about this particular kind of macromolecule when we get to our unit on genetics and heredity later on in the year.